The study of temperature in physiology is important because a significant drop or rise in temperature can affect the way our bodies perform. A fever results in an elevated temperature in the body and can lead to symptoms such as chills, sweating, and weakness that occur as a result of the disruption of the body's homeostasis. Cold temperatures lead to symptoms like shivers, depressed heart rate, and muscle stiffness. Today we will share with you the effects of temperature on the sciatic nerve of a frog. The sciatic nerve is used to demonstrate the ways in which neural processes and conduction changes with varied temperature. The purpose of our experiment was to examine the effects the different temperatures of Ringer's solution would have on the threshold and recruitment, conduction velocity, and refractory period of a frog's sciatic nerve. We used Ringer's solution at 25, 15, 37, and 60 degrees Celsius. Before we go too into depth on the results we obtained, here is a little background information that will help you understand how the sciatic nerve works. Within nerves, there are A, B, and C fiber types. The frog sciatic nerve contains mainly A fibers. Among the A fibers, there are alpha, beta, gamma, and delta fiber subtypes. Of these, A alpha fibers have the lowest threshold, and therefore are the first to fire when the nerve threshold is reached. A beta have a higher threshold, and A gamma have an even higher threshold to be recruited. A delta fibers have the highest threshold and are the last of A-type fibers to be recruited. The summation of all the fibers, alpha, beta, gamma, and delta, result in a compound action potential, or a CAP. A single action potential is generated when a neuron is first stimulated. The stimulation causes sodium channels to open, allowing sodium to travel down its electrochemical gradient. The entrance of sodium into the cell causes the membrane potential to increase. When the membrane potential reaches threshold, voltage-gated sodium channels become activated, causing a depolarization initiating the action potential. As opposed to an action potential, which is an all-or-nothing event, a CAP is the sum of individual action potentials that multiple fibers can conduct. In our experiment, we analyzed the latency. This was calculated from the start of the stimulus amplitude to the peak of the CAP. We also looked at the amplitude of the CAP, which is measured from the baseline to the peak of the positive or negative deflections. The experiments were each performed with a set of 4 temperature conditions in this order: room temperature, 25 degrees Celsius, cold, 15 degrees Celsius, warm, 37 degrees Celsius, and hot, 60 degrees Celsius. These temperature conditions were administered using Ringer's solution that was warmed or iced to the desired temperature. The treatments were administered by filling the nerve chamber with Ringer's solution of the appropriate temperature. The nerve was then soaked before taking extracellular recordings. The threshold and recruitment, conduction velocity, and refractory period of the frog sciatic nerve was then determined for each temperature.
To measure the threshold and recruitment of the sciatic nerve, the nerve was exposed to gradually increasing stimuli to determine the threshold voltage required to initiate a CAP, as well as the voltage at which the maximum CAP is reached. The stimulus was first set to an amplitude below threshold on the PowerLab stimulator. We used a value of 40 millivolts. The amplitude was then increased by 5 millivolt increments until a threshold was reached and a CAP was observed. After threshold was reached, the stimulus was increased by increments of 25 millivolts until a max CAP was obtained. We recorded a few more CAPs with increasing voltage past the maximum just to confirm that the CAP did not continue to increase in size. We repeated these steps for each temperature condition and observed these results. For the room temperature Ringer's solution, the threshold was found to be 60 millivolts and a max CAP amplitude obtained at 300 millivolts. At a cold temperature, 15 degrees Celsius, we saw an increase in the threshold and in the stimulus required to reach a max cap. The threshold was found to be 70 millivolts and the max cap was reached at 390 millivolts. At the warm temperature of 37 degrees, we saw a decrease in both the threshold and in the stimulus required to produce a max cap. The threshold was at 55 millivolts, and we reached max cap with a stimulus of 225 millivolts. The nerve and hot ringer solution produced no recording for the threshold in recruitment because the nerve could not generate any action potential. This was a result of a temperature out of the physiologically favorable range. This resulted in a loss of function in the sciatic nerve. The major findings in this experiment showed that as temperature increased, the threshold and maximum cap decreased. The exception is for the trial of 60 degrees Celsius, since no nerve function was observed. The mechanism for this observation is largely due to membrane fluidity. A decrease in the temperature causes the lipids around the gating mechanism of voltage-gated sodium channels to decrease in fluidity, therefore causing a decrease in sodium conductance. In other words, the decrease in membrane fluidity caused the sodium channels to open less efficiently and made the sodium channels less accessible. Similarly, an increase in temperature increased the membrane fluidity, allowing the voltage-gated sodium channels to be more accessible, leading to a greater sodium influx.
Conduction velocity measures how fast an action potential propagates through a nerve. To measure the A-alpha conduction velocity, caps were recorded with the negative recording electrode at position D and at position E. The latencies of the two caps were then used to calculate the conduction velocity. We repeated this experiment for each temperature condition. Conduction velocity is calculated by taking the distance traveled and dividing it by the amount of time it took to travel that distance. The distance between recording electrodes D and E was 10 millimeters. The example shown here is the conduction velocity of the control, which was the 25 degrees Celsius Ringer's solution. The conduction velocity for room temperature Ringers was found to be 62.5 meters per second. For the cold temperature, the conduction velocity was calculated at 45.45 meters per second. At the warm temperature, 37 degrees Celsius, conduction velocity was calculated to be 52.63 meters per second. At 60 degrees Celsius, there was no nerve activity detected. Conduction velocity was recorded as an indicator of how quickly the nerve is able to propagate electrical signals. Based on our results, the general relationship between conduction velocity and the temperature were as follows. At colder temperatures, we observed a decreased conduction velocity compared to the control and warmer trials. At warmer temperatures, we observed a faster conduction velocity than the colder trial. The changes in cap conductance speed observed for variable temperatures conditions are explained by the temperature dependence of sodium channel activation. At lower temperatures, the lipids controlling the opening of the voltage-gated sodium channel gates are inhibited. This results in a slower opening of the sodium channels and ultimately slower conductance of sodium. This elongates the rising phase of the action potential and therefore decreases the conduction velocity. The opposite physiological response occurs at warmer temperatures. At warmer temperatures, the lipids controlling the opening of the voltage-gated sodium channel gate are more fluid. This results in a faster opening of the sodium channel and ultimately faster conductance of sodium. Our results also demonstrate the significance of physiologically favorable temperature conditions. Conduction velocity was faster at the warmer conditions versus the cold condition. However, the room temperature conduction velocity was the fastest. This is due to room temperature being a favorable temperature condition for a frog. As temperature became too high, loss of function of conductivity occurred. At 60 degrees, no nerve activity was observed.
The refractory period indicates maximum frequency at which action potentials can fire within a neuron. There are two stages within the refractory period, the absolute and the relative. During an action potential, the neuron is said to be in the absolute refractory period, where no matter the stimulus magnitude, no action potential can be generated due to the voltage-gated sodium channels closing their inactivation gates. The absolute refractory period is over once the sodium channels open their inactivation gates and allow sodium to flow through the channel during the next action potential. Directly following an action potential, the neuron enters the relative refractory period while in its repolarization and hyperpolarization phase. During this time, another action potential can only be generated if a stimulus of a greater magnitude is fired. A greater stimulus is needed to combat the potassium leaving the cell and driving the membrane potential even more below the resting membrane potential and the value of the threshold. To measure the refractory period, the power lab stimulator was set to double to initiate twin pulses. The first recording was taken at an interval of 9 milliseconds so that the max caps were produced consecutively. The interval was decreased by 1 millisecond for each recording up until 4 milliseconds. After this, the interval started to be decreased by 0.5 milliseconds. The above steps were repeated for each temperature condition in the same order as previously mentioned in the treatment procedures section. At room temperature, absolute refractory was reached at 0.75 milliseconds. At 15 degrees Celsius, absolute refractory was reached at 1.25 milliseconds, and at 37 degrees Celsius, the absolute refractory was reached at 0.75 milliseconds. At 60 degrees Celsius, no nerve activity was observed. Ultimately, the refractory period for the room temperature and warm temperature was shorter than that of the cold temperature. The refractory period for the room temperature and warm temperature was shorter than that of the cold temperature due to temperature altering the rate of ion channel gating kinetics. At temperatures of 25 degrees Celsius and 37 degrees Celsius, there was a shorter refractory period due to quicker opening and closing of the inactivation gate. At the cold temperature of 15 degrees Celsius, the opposite occurs. At lower temperatures, the sodium channels remain open longer due to slower movement of the inactivation gate. In this experiment, a frog sciatic nerve preparation was used to study the physiology of neural compound action potentials in response to a variable temperature. The results of these experiments demonstrate that the neurophysiology in frogs is a useful means of studying concepts related to other species. However, the range of temperatures used in this experiment demonstrate the physiological significance of temperature within different species. The results indicate that temperature significantly influences neural function at extreme temperatures. The effects of temperature observed in these experiments result from the membrane fluidity and sodium channel activity. Membrane fluidity increases with temperature. This leads to the recruitment of voltage-gated sodium channels. As a result, at higher temperatures, neural function exhibits a lower threshold, faster conduction velocity, and shorter refractory period. Colder temperatures induce depressed nerve activity. But in relation to our results, these trends only hold under physiologically favorable conditions that are species-specific. At significantly high or low temperatures, the nerve demonstrates a loss of function.
Therefore, these findings are better explained by the temperature dependence of sodium channel activation and inactivation than by lipid membrane dynamics. These channels prove to have a high sensitivity to being inactivated while at high temperatures.